So now let me introduce Dr. Angela Kranz. So Angela studied biology at the University of Mainz before completing her doctoral degree at Forschungszentrum Jülich, Heinrich-Heine University in Düsseldorf in Germany at the Institute of Bio and Geosciences, Biotechnology, IBG1. During her time there, Dr. Kranz analyzed genome and transcriptome data to characterize the bacterium Gluconobacter oxidans. As a postdoctoral fellow in the group of Professor Bott at the IBG1, she manually curated a large set of expression data stored in an in-house microarray database. And since 2020, Dr. Kranz has been working as a scientific coordinator at IBG4, Bioinformatics, Forschungszentrum Jülich, and NFDI for the NFDI project DataPLANT. So over to you, Angela. I'm going to stop sharing my screen and you can start sharing yours.
Yeah, thank you very much. Can you see my screen?
Yes, I can. Thank you.
Good. Thank you. Okay. Then let's start. Yeah, thank you very much, Marie, for the kind introduction, and of course also for the invitation, also to Elizabeth, who is sadly not here today. So today I want to present you the project DataPLANT and focus a little bit more on our ontology service landscape. I will briefly introduce to you the NFDI DataPLANT, will then come a little bit to our main concept, the annotated research context, will furthermore introduce our metadata toolchain, which includes the mentioned ontology service landscape. Kind of a little bit to DataPLANT. DataPLANT is a German NFDI. NFDI stands for National Research Data Infrastructure. The main aim of the NFDI is to systematically manage scientific and research data, provide long-term data storage, backup, and accessibility, and network the data both nationally and internationally. At the moment, there are 26 consortia embedded within the NFDI. Actually, at the moment, the first NFDI conference takes place here in Germany in Karlsruhe. The consortia are quite diverse with respect to the different domains they tackle. They span the humanities and social sciences, engineering sciences, natural sciences, and life sciences. And all of them have one united common purpose: to develop sustainable solutions for fair data management. Furthermore, there is a collaborative effort called Base for NFDI, which exists within the NFDI landscape. This brings together all consortia to harmonize their strengths and also their purposes in order to develop fundamental services there that can be applied for all different kinds of scientific disciplines. So DataPLANT, as the name already suggests a little bit, focuses on plant sciences, specifically more on the fundamental plant sciences, and aims at providing a flexible tool chain and also standards, services, and so on to structure research data according to the FAIR principles. The main target group of DataPLANT are mainly researchers who work daily, for example, in the lab, in the greenhouse, on the field, and who also do data analysis. The DataPLANT team consists roughly of around 50 people. And we also have many associated members, which helps us with, for example, data stewards or also testers that test our software, our services, and the infrastructure. The main task areas within DataPLANT are four. The whole DataPLANT consortium is led by Dr. Dirk von Suchodolitz, who works at the University of Freiburg. His main target group are infrastructure and also service people. At the University of Düsseldorf, Dr. Jens Krüger leads the task area that mainly focuses on software development, services, and also infrastructure improvement. At the University of Kaiserslautern, the task area lead is Professor Dr. Timo Mühlhaus. Here within DataPLANT, this task area focuses on transfer, application, and education to also facilitate training of our tools and services to the broader audience. Furthermore, the annotated research context was developed by Professor Mühlhaus and also several softwares. We in Jülich mainly focuses on standardization, quality, and also interoperability. The task area is led by our head of institute, Professor Dr. Björn Usade. So as most of you know, effective research data management is paramount, and it should adhere to the FAIR principles in order to make data findable, accessible, interoperable, and reusable. Undoubtedly, if researchers want to adhere to these established principles and also guidelines, such as the FAIR principles to make the data in the end fair, demands usually a significant investment of also effort and energy to the researcher when it comes to managing research data, especially, of course, for the inexperienced users, PhD students who may be just starting their project. Data management, of course, comes together with new responsibilities for the researchers. There are roughly around 600 different kinds of content standards that are available, who somehow refer to methods, formats, terminologies, and also guidelines that help with the research data management. Also, usually a user has to think about checklists and also repositories that they target when they want to publish their data. Consequently, based on this huge amount of different kind of guidelines, principles, standards, users often find themselves faced with the challenge of navigating this extensive landscape to determine which specific guidance might be of importance for them or what they should prioritize. The aim of DataPLANT is to reduce this kind of energy that is required to make data fair by providing a specific set of tools and services. Particularly, if we speak about diverse omics data types, such as, for example, genomics, transcriptomics, or metabolomics, one important step during publication is also the upload of the data to the endpoint repositories, such as GEO, ENA, or metabolites, in order to meet the requirements that the repositories have in order to upload the data. It is usually imperative to adhere to different kinds of checklists and also to use controlled vocabulary and standards. And these might also often vary between the different kind of repositories that are available. And of course, data management also is not just important for the raw data, but also encompasses process data resulting from the sophisticated analysis pipelines, like, for example, using tools such as R, Galaxy, or Nextflow. The annotated research context, which I mentioned, actually offers all this together and functions therefore as a single point of entry logic for data management and also for computations. In a straightforward manner, the Annotated Research Context, which is shortly called ARC, serves as a container for standardized and structured storage of experimental data. But not just the experimental data, but also computational analysis steps, as well as the annotation of the data. Data that can be stored in an ARC is quite diverse. So each kind of data that one can imagine can actually be stored, like ranging from, for example, phenomics data, proteomics data, and also genomics data. Delving a little bit more into further detail, the Annotated Research Context is actually a structured folder system, which is designed to provide a user-friendly framework for organizing the research data and the analysis data. It includes by itself a versioning system ensuring data provenance and data integrity. The folder structure can be divided into several subfolders, catering to various facets of the investigation. Specifically, they are designed, for example, subfolders for describing the sample material data, the measurement data, which might be, for example, the sequencing data from omics or transcriptomics experiments, and also dedicated folders for housing the used analysis workflows and their corresponding results. The ARC itself can be described as a FAIR digital object adhering to established standards in the field. Its folder structures, as you might have already noticed, mirrors the ISA standard, featuring the investigation, study, and assay layer within the framework. The investigation layer serves to elucidate the fundamental context of an entire experiment within an ARC, while the study layer encompasses detailed information, for example, about the study design, which might be the used plant species, or also the different kind of treatments that a plant was treated with. On the other hand, we have the assay folder, which houses critical details leading up to the data files itself, like, for example, DNA isolation, the library preparation for sequencing, and also the sequencing itself. For describing the computational aspects of an analysis within the ARCs, in order to standardize also these and facilitating also reproducibility for different kind of analysis, the common workflow language is employed at this step. Each layer of the folder structure is then complemented by its corresponding metadata. In this case, they are variable, as you can see, as Excel files, but the metadata is actually available then in the ISA tab format and can be easily transformed to other ISA formats. To assist with the metadata annotation, DataPLANT provides the Suite Excel add-in tool. This tool is available for the local Excel version, the online version, and also recently we have a first version of a completely standalone version, so that also users who are not using Excel have access to Suite. Suite itself facilitates a swift and efficient method for annotating the study and assay metadata sheets with the respective data in the folders. It adopts a simple spreadsheet-based approach, offering per column-wise different kind of building blocks to describe respective metadata. These metadata are described in a parent-child term relationship. To collect metadata, Swade leverages ontologies in order to standardize both the column header and also the values within the table. So for example, in this case, we can see that one of the metadata that was collected was the protocol type. This was added a specific building block within the spreadsheet and can then be filled out to collect the respective metadata. So, and here you can see how it's further used. So using ontology term search, the respective value to describe the metadata for a specific sample can be found via the ontology term search. In this example, for the growth protocol was selected as a protocol type that was used here and was then used to fill out the respective row, and each row can here represent a different sample that was annotated with respect to the different kind of metadata. In order to fulfill specific requirements that might be desired or also that are necessary if people want to upload the data later on to specific repositories such as HDO, or if the metadata that should be collected should fulfill minimum information standards. DataPLANT already has a certain amount of templates provided within the Excel tool. They can be accessed via a database and directly selected from it. So they are actually curated by our data stewards and also by some developers. And the templates can then directly be loaded into Excel and then be used to collect the different kind of metadata with respect to the experiment that was used. So to elucidate the comprehensive suite of services and tools that are actually offered by DataPLANT that are designed to streamline the metadata annotation and ensure standardized research data management, I want to explain to you now the metadata toolchain within the DataPLANT infrastructure. At the core of this toolchain, we actually find the arc that I just explained to you. In order to create these folder structures, there is a software available, which is called arc commander. The arc itself can be exported, for example, also as a JSON file, and the metadata, also the data, can be converted in such a way that the metadata alongside the data can easily be uploaded to the different kind of repositories. Also, it is possible to store the arc on our own DataPLANT data hub. And the arcs that are published there, they can be kept private just for its own use. They can be shared with colleagues or whole teams, but they can also be made publicly available and can get enjoyed. Actually, our first arc was recently published with DOI. So on the other part of the metadata tool chain, I will focus more now on is our ontology service landscape, which is a crucial component that supports the metadata annotation through the Swade tool. So as I explained already, metadata annotation can be achieved through the Swade tool. and this is actually achieved via the SWAIT ontology database, which is a Neo4j database. The ontology database remains seamlessly updated through the so-called swap-up service, which actively retrieves data from a GitHub repository, which hosts a broad range of different kind of external ontologies, as well as the DataPLANT biology ontology. In the upcoming slides, I will now explain to you a little bit more in detail the different components. So in this segment, let's dive deeper into the swap-up tool. Swap-up serves as the link bridging between the SWAIT templates and the ontology and the SWAIT database. Its operation is reliant on a GitHub repository, which allows data integrity through file versioning. SwapUp recalls the ontologies to a Neo4j database here, and is managed how nodes and so different kind of ontologies and also our DataPLANT ontology are connected with each other. And SWAIT is able to directly read from this database to allow the ontology term search and also to allow seamlessly annotation with respect to the child-parent term relationship via the SWAIT tool. So the SWAIT database is a repository that has different kind of external ontologies stored within. And all these external ontologies are at the moment essential for proficient metadata annotation. As you can see here, they are quite diverse. So we not just only have ontologies related to plant sciences, but they also span the biology and chemistry field and also experimental fields. So for example, with respect to different kind of plant focused ontologies, we have the plant experimental and conditions ontology, plant trait ontology, and the plant ontology. And with respect to, for example, describing a different kind of experiments, we have, for example, the experimental factor ontology, and also ontologies such as Chebi or the mass spectrometry ontology. The selection of these ontologies was made in close collaboration with the scientific community at the beginning of the development of SWAIT, taking into account the ontologies commonly employed in annotating the metadata, and also not just for the fundamental plant science experiments, but also with respect to the different kind of omics technologies that are used. And also, of course, we have in the SWAIT DB our DataPLANT biology ontology, which I will explain later. It's worth noting here that the collection we have here can be effortlessly customized to also adjust the needs of other research domains, for example. As previously told, our collection of the external ontologies we have and also our DataPLANT biology ontology are openly available on our GitHub repository. As previously mentioned, the DataPLANT biology ontology serves also as a source of missing terms that can be used to complete their associated relationship to pre-existing terms, which are essentially for the user that need them to annotate data with a SWAIT tool. Much like the process that is already established for many different kinds of ontologies that are published on GitHub, our users have the opportunity to actively engage in shaping and improving this resource, they can do so by, for example, submitting issues, suggesting, for example, new terms, and also import terms from external ontologies, or improve already existing terms by adding, for example, relationships to already available terms. In case where users identify the need that they need to have another external ontologies to describe their experiments via the SWAIT tool, a request can also be made that they are added to the SWAIT database. This typically occurs in cases when over 60% of the terms from a specific ontology are essential for the metadata annotation according to the user that is actively working on annotation with SWAIT. For those who prefer not to utilize GitHub issues, an alternative is also to submit a ticket via our help desk at DataPLANT. So following the input from our user team, the DataPLANT ontology team proceeds then after an issue is submitted to integrate the suggested terms into the DataPLANT biology ontology. These additions are then, of course, carefully integrated into the ontology by adding, for example, also relationship in form of SA. This process can be exemplified, for example, here where the plant growth protocol I mentioned before as an example within the SWAIT template was integrated into our DataPLANT biology ontology as part of protocol type, saying that the plant growth protocol is a growth protocol, which is then a protocol type and can be added to the respective building block in the SWAIT template. So usually each term that is added and assigned by a specific DataPLANT team member is directly weighable then within SWAIT for metadata annotation. Furthermore, the DataPLANT team is actively engaged in bridging the ontology gap that is experienced by the users who require specific terms for the data annotation. This facilitates the exchange of knowledge between the researcher and then also the main ontology provider. In this role, DataPLANT acts kind of a broker by suggesting the terms that our users have suggested to us back to the relevant ontology providers, thereby also improving these external ontologies. And also this not just enhances the usability, but also reinforces the integrity of the entire ontology landscape that is used. So with this, I am at the end of my talk and briefly I'd like to summarize what I just presented to you. So DataPLANT is an NFDI which offers a broad range of tools and services that can be used for data management. Within DataPLANT, we have our DataPLANT ontology service team, which assists with metadata annotations via the tools SWAIT and SWOOP-UP. We also have our own DataPLANT biology ontology, which can be used to add terms that are missing by the users for metadata annotation within SWAIT. And all the concepts that I just presented to you can be easily adapted also to other research domain and extended. So this brings me, of course, to the acknowledgement. So DataPLANT is a huge consortium, and all of this was done by a great team together. Special thanks goes out here to our team at the Forschungszentrum, especially to Catherine Dumschott, Hannah Dörpels, and Björn Osade. Catherine and Hannah are the main people working on our ontology service. Within the group of Timo Mühlhaus at Kaiserslautern, special thanks goes out to Lukas Weil and Kevin Frey, who are the developers of the ARC and also the SWAIT tool, and also to Dirk von Sochodolets and his team member Marcel Schöpel, who is responsible for developing the SWAIT database and also the swoop-up tool, and also, of course, to all the other members who are part of DataPLANT, who help with the testing and who are always there for any kind of help if there is a problem with a service or software. This was actually during our last hackathon in Jülich. And of course, I would also like to thank you for your attention, and I am open for any questions. Yeah, and I forgot, of course, different kind of information about DataPLANT can be found on our homepage. All tools I just showed to you are at GitHub, and we are also present on Twitter or X. Thank you very much.
Thank you, Angela, for the presentation. That was very, I mean, it's great to see the work that you did trying to bring all these different pieces together. Now we'll open it up for questions. So there's no question in the chat box. So is anyone wants to add a question? You can just unmute yourself and just ask the question. Yes, Pavel, do you want to ask something?
Yes, if possible. Thank you very much for this presentation. And okay, it's a new tool for me, so I'm just looking at the GitHub webpage of yours, and I'm looking through the list of templates. So you don't seem to have one for phenotyping?
We actually have, so
somewhere else.
Yeah, so we have one for phenotyping, and it's actually MyAP compliant, but somehow it crashed our database lately when we wanted to add it again. So at the moment we just removed it again. I'm not sure why. So there's nothing to do with phenotyping, of course.
Okay, thank you. But I also understand that the whole GitHub site is a collection of tools and let's say templates. So what does the user have to install some software also?
Yeah. So the SWAIT tool, like I said, it's an Excel add-in which can be installed, and also the ARC commander to generate the ARCs also needs to be installed. But at the moment, we are also working. So the ARC commander is at the moment Linux, not Linux-based, but command line based. But at the moment, we also have a new tool, which is called the Architect, which is in its first testing phase. And here we have a nice GUI where you can directly generate your ARC, and later on, you will also have the possibility to annotate your metadata in this tool. So just that you just need to install one software for all in the end.
Thank you very much.
Thank you, Pavel, for the question. So I see that there is a question by James in the chat. James, would you like to ask the question live, or should I read it? Yeah, okay, I'm going to read it. So, Angela, you mentioned that ARC uses a versioning system. Is this a bespoke system developed for ARC, or does it leverage existing version control system like Git?
Yeah, thanks for the question. It uses existing version control system via our GitLab repository.
Thank you. So does anyone has another question for Angela? If not, I have one, Angela. So you mentioned that for the DataPLANT biology ontology, you are creating terms that are missing, and then you are submitting terms to the external ontologies. You know, you act as a broker. So when the external ontology creates the term, what will happen in your system? Are you gonna deprecate the term in the biology ontology, or what's the plan?
Yeah, we will deprecate it.
Yeah, okay.
Yeah.
That's good. That's I mean, that's a huge curation work that's needed, and I agree that the way to not be stuck in the process. So that's a really good way of doing it, and that's great if you have like curators that can do that. Any other questions, comments for Angela? If not, I'm gonna just close the meeting. Can you stop sharing? So so let me share my screen quickly, just to PowerPoint. So that was the presentation. So the Q&A is done. So so if you have any question regarding the community of practice, please contact Elisabeth Arnaud. She will help you with whatever you ask her. And so, thank you all for joining us today. Thank you, Angela, again, for the great presentation. I mean, I'm very excited to see like how those arcs can work, and also how they can help to build better ontologies. So thank you all. And we will probably have like a webinar next month. So we'll keep you informed. Thank you very much. Bye-bye.
Thank you.
Very much.
Bye.
Bye.
Thank you. Bye.
Bye.